Oh.
Well, you're close.
Am I not going to see a moose down here?
Moose octopus.
Must be getting close to 4 o'clock.
Moose octopus. Anything other than a moose?
Good save.
Those deep sea moose are hard to come by.
And he's the moose octopus or something like
Yeah.
this.
Right. That's the closest moose we're going to get, I think. I don't know, whenever we're doing blue water, I'm always just like hoping to see something.
Like a whale.
Like a whale,
Yeah,
exactly. I mean, just something really, you know, just.
Or a whale fall. I mean, that would be, you mentioned that earlier.
Cool. Yes, that would be really neat.
Especially because we're over an area that does have, like you were saying earlier, these are more productive sometimes over the seamounts. There's evidence for that. So you might expect to have a whale fall.
We do have some issues along portions of California too with, unfortunately, large vessels running into whales. And so it's possible sometimes those whale carcasses that you come upon are there because they were injured or killed by interactions with large vessels. We're a lot of work. Sanctuaries are partnering with a lot of others to try to reduce the incidence of that, but that can happen.
Go ahead and push on in their video for a minute.
We also had
quite a male, I think.
Quite a few gray whales. There was an unusual mortality event for gray whales this year, and so some of those whales had to have died by sinking down, as some have washed ashore, but there have to be others that did sink and fall to the seafloor.
All right, go ahead and cut my video.
Yeah, it's tucked away, so
Thanks.
likely a male, I'd say.
Oh, we have a ways to go, eh?
Yeah.
Do you think we'll get there before the end of our watch?
Maybe.
I hope. That would be great.
What do you think, Aaron?
The very tail end of it, maybe.
Be close, I guess.
Yeah, close.
Put the pedal to the metal.
Pilots, do you want a speed increase, or are you happy with this?
Let's see.
What's it doing now?
It's 0.3.
Yeah, attempting 0.3, so 0.2 to 0.4.
Pretty steep. Let's just wait another few minutes till we get on top of this bit of ridge line here where it flattens out.
Okay.
And then we can try and do that.
Science, do you expect that we'll be wanting to zoom on items of interest, or is that just a pastime?
If it works, but I'd rather get to the transect.
Right.
Than to go slow just to be able to zoom in on things.
Sure.
So
yeah, okay.
we do have a mushroom coral right there. You can see the pink
Yeah, go
circle
ahead.
with the
Call it in now,
stalks.
Aaron. It's not very steep. Herc is
Yeah.
almost over that.
Right. As much as
0.5 or something.
0.5. All right. Bridge nav.
So what we've decided to do right now is
Can we increase speed to 0.5 knots?
Increase
Thank
the
you.
speed of the ship so that we can move between the transect we just finished and the transect we want to start a little more rapidly. May mean that we'll see something interesting and we won't have time to stop and take a closer look, but I think we'd rather get onto that next transect, continue our progress in surveying the distribution of octopus and other organisms on this feature.
All right, I'll stay out here, Mike.
Is that an octopus way off to the left, maybe?
Unconfirmed.
Which side? Oh,
Left.
up left.
Yeah.
Looks like it. No, it's a Cuskill.
Cuskill. Okay. Thank you for looking.
Sure.
Oh, beautiful anemone. Can we, as we pass by, can we just get a little bit of a more of a zoom on that anemone, the purplish black one?
Yeah, sure. 1 sec. All right, video, go ahead.
Wow.
Thank you.
Yeah, sure. Yeah, better angle
Oh, there.
here.
There you go. Beautiful.
Yeah.
Mike's.
That's
Mike,
a good
someone
one.
writes in, poor Mongo gets neglected. Has it even been used this month?
Nope.
No,
Mongo?
he was fundamental in placing the Osmo sampler.
All right, go ahead and come my video.
Was that about the
Mongo?
Mongo worm?
Yeah, he held the he held it so that we could remove the T handles because otherwise the Osmo
Oh,
sampler
yeah.
got tipped.
All
I'd
right.
like to use Mongo.
Bobby
very-
from Burbank would
Very
like to
essential
know
to the operation.
what kind of an upgrade would Mongo need to be more likable?
Replacement.
Oh. What are you saying?
Mongo needs to be replaced.
Needs to go into the Nautilus Museum of obsolete parts.
Yeah.
No love
No,
for
it serves its
Mongo.
purpose, I guess.
Mongo strong, Mongo good.
So as we're going along, you might see some kind of brown masses on the seafloor. We've been talking about those earlier today. Xenophyrophores. I think I said that right.
Yep.
Yep.
What's the wind? 20.
It's been about 18 to 20.
That was a good little hiss from the
Yeah.
ship.
So that black coral's missing some sections there, but
Does that happen when another organism climbs up it, or does it
It
maybe just get hit in passing?
it can. Sometimes you'll see like a star kind of wrapped around a portion of it. That could, I guess there's other things that could run into it, damage it, and break off areas. They appear to be able to survive with some portions of them missing. And I don't know about regenerate tissue over areas that have lost tissue.
Do you know if these kind of different variety of sea stars that we've been seeing on the seafloor, are they able to regenerate like the ones that you may find at the New England beaches are? Like one
I
of their
would
legs gets
assume
chopped?
so. That if they get cut up, they can regenerate as long as there's a portion of the central disc and arm. They can regenerate. But we haven't really seen much evidence of that as far as they all seem to be fairly intact.
Yes.
Here, this octopus is actively walking
He is.
across the seafloor.
It is.
This octopus is actively moving, but
Kinda
I'm not
cruising
seeing
along
it's
there.
using that third arm. It's not extended the way the others are.
Did you want to look at it real quick?
I don't think we'll be able to see the third arm,
Wait.
so I think we can just continue on.
Sure thing.
Thank you.
Once we start coming up to speed and feeling that speed, it'll be a little bit faster video for you guys. So it might be a little harder to stop and zoom on stuff.
They might never get up there.
Yeah.
It's doing pretty solid. I mean, it fluctuates, but it's HIPAC's been 0.6, 0.7 every time I look at it.
There's one of your tulips on the left.
Yeah, that
Very
one looks
tulip-esque.
very tulipy. That's a sponge, but looks like a tulip.
Yeah, all of these things we're seeing, they're all organisms, right?
Hmm?
We're seeing organisms.
Yeah.
All of these plant-looking.
Oh yes, they're not plants.
Yes,
That's
they're
right. They're all
organisms.
animals. There are no plants down here. 'Cause there's no sunlight. So even though they look plant-like, that's right. They're all animals.
Well,
Oh yeah, you're right. Plant is an organism.
although it's interesting because the protist is a primary producer, is it not? That xenophyophore?
Well,
No,
no, it's a consumer, right? It's consuming the detritus in the water column.
Yeah.
Right, so in the deep sea, the only primary producers we have, so those are animals that are taking, you know, at the surface they're using sunlight as their energy source to make food. In the deep sea, animals that can convert energy to kind of start the food web would be ones that are
Chemoautotrophs.
exactly. They're using chemicals that are seeping out of the seafloor, and they can convert those into sugars.
and the seeps here, we should say that it's not the same chemicals that we
No.
see. Those would typically be methane seeps. And as far as I understand, they haven't measured that type of seepage in this zone.
Right. And some of the other types of animals that you see that are capable of that, sometimes they're tube worms or bacterial mats or a certain kind of clam. We haven't seen any of those animals either. That can sometimes be your clue that you're in an area where they're seeping methane. And we have seen that on Davidson Seamount proper, a little area where there are these vesicomyid clam shells, and that was very interesting. Or wait, was that on Sur Ridge? Oh, now I can't remember.
But it's along in the Pacific, California coastal.
Yeah, we do have cold seeps
region, yeah.
in Monterey Bay National Marine Sanctuary. Just not, we haven't seen any evidence of them here. So a cold seep's different than some of those like hot smokers or other kind of really warm areas.
The inside of the Earth is very
here
dynamic, and so we have a lot of different types of venting, a lot of different variety of chemistry.
It's neat. There's some animals that are seen on whale falls that are able to take the fat that is stored inside the bones of the whales and use that as their energy source. That's pretty cool.
How long does a whale fall typically last? I know a community will establish itself, and then
there's succession.
Yeah.
So you get a fresh whale fall, you'll get a lot of animals like sharks and fish, hagfish, your favorites will come, and they're going to feed on the flesh that's on the animal. And then you'll have some of the slower consumers and decomposers like the crabs and the shrimp, and all these animals are going to come in and kind of clean the skeleton. But as I was saying, there's a lot of fat and oil stored inside the skeleton itself, and so then these other animals that are like worms and other things will then establish and be on the skeleton and use up all the rest of the resources that are there. So it's decades that actually a whale fall can persist in feeding some component of the deep sea. And you don't see these worms anywhere else. They're
very specific to the whale falls?
Yeah.
That's really fascinating.
So some of the things that we probably see floating around here are not just marine snow falling down, but also larval forms of deep sea animals that are moving amongst these patches of hard bottom, or these patches of, they probably can occur on things other than just whale falls. I mean, other animals, large animals die out here and fall to the seafloor. So, but from those kind of large patches of food falls. And we
Can
found
can we stop tracking the line and move to steps, please?
one interesting thing we found in Monterey Sanctuary was a shipping container that had fallen off of a large, shipping transport. It was unexpectedly found, but then through research about the source of that container, it was determined that
Bridge nav.
a number of other containers had also fallen
can hold
off
position
and would
there.
likely
Thank
be
you.
on the seafloor in the sanctuary. And so here's a artificial hard bottom that could serve as stepping stones for some organisms to move around as well. Hopefully mostly native, but there's potential that they can also serve the purpose of allowing non-native species to spread around. So kind of interesting.
Do you guys see a lot of evidence of invasive species in the sanctuary?
We do have some in shallower waters, like the estuary that's in the sanctuary called Elkhorn Slough has quite a few different non-native or not just non-native, but invasive species, and some algae in the rock intertidal and kelp forests and harbors. We have some of that, but we don't know of invasive species in deeper waters. But of course, we haven't fully characterized the flora or the fauna of the deep sea, so
I imagine it might be difficult to, in terms of the source of a deep sea invasive and what the mechanism for it getting transported to another site.
Mm-hmm.
You know, with ships and ballast water and things like that, shallower organisms, the potential for invasion is much higher, I would imagine.
Yeah.
Oh, sorry.
Again, why it's so important to explore these deep areas and try and get baseline information on what is there.
Yes. We don't have a full grasp of the diversity, the species diversity that we have in these deep sea areas, so it's really impossible to tell if it's changing.
Kind of going back to the shallow area for a second while we're on this blue water transit, what kind of steps would they take to mitigate invasive species in the shallower areas?
Well, it kind of depends on how extensive the invasion is. So we were seeing this one algae that was mostly in harbors, and so there were divers going out trying to actively remove it.
Is that the Unundaria?
Undaria, yes.
Yeah.
But it's hard to maintain that kind of a removal program. It's pretty labor intensive. And you have to be careful. Sometimes invasive algae like to be kind of fragmented and can spread through fragmentation. So you kind of have to be careful.
Yeah, ecology of weeds. We had a
Barely hear you, Jess.
they had a
Speak up.
red abalone that was from South Africa, not the same red abalone we have, but they tried to introduce it to some of the abalone farms, and it had a sabellid worm. And they were very concerned about the invasion because these abalone farms are right on the coast, and so they use seawater, pump live seawater in and back out. But they caught it so early. Dr. Armin Kuris, who's a parasitologist at UCSB, and an army of graduate and undergraduate students went to every farm where there was effluent, and they collected and checked every marine snail. There was a potential host for this, and it seems to have been eradicated.
Wow.
Great.
That was a couple of decades ago now. But yeah, just talking about different ways that these invasives can get to us and what we can do about them.
Right.
So many times it isn't until we see them, you know, in significant numbers, and then it's very hard to eradicate them because they can transfer so far
Right.
through
So
ocean currents.
yeah, the ones that establish themselves in harbors, often they're growing on maybe the piers and pilings, but then they can also grow on the hulls of vessels, and if some vessels are not regularly cleaned and defouled, they can then serve as transport to bring them from one harbor to another.
Science, I'm going to lose visual contact with the ground for a minute.
Okay.
Okay.
In bringing it back to the ship, actually, when I got on the other day, I noticed that there was a power washer
Come down a bit there,
on
Mike.
Argus.
When you get room on the sonar.
And so they were power washing Argus because it was
5
in the
meters.
Cordell Banks Sanctuary, and they wanted to make sure that there were no hitchhikers that could potentially wind up in the Monterey Sanctuary. So they give it a blast.
That's great.
Yeah.
Yeah, there is another algae. It's a sargassum.
Oh, yeah.
And it is in Channel Islands National Marine Sanctuary, and as of right now, we don't have it established in Monterey. We did have a couple sightings of it on the beaches, but just as wrack, and we don't know its source. And so divers went out and tried to determine if it was in the kelp forest in the surrounding areas and didn't find it. So we're definitely trying to not have it establish itself in the kelp forests in Monterey Sanctuary.
Yeah, so I've been free diving and scuba diving out at Catalina Island since I was a 4-year-old. And I've seen it, the sargassum come, and it's everywhere out there now. Just terrible.
It's all over the East Coast as well. One of the graduate students in our lab is doing a study on sargassum and trying to do predictive habitat modeling to try to understand where else they are and then where else they are potentially going to be. And he's been able to find data sets from all over the Atlantic Ocean, particularly around the coastal regions of the northern
This summer I was
areas.
in Belize, and they were complaining. I was talking to a dive operation, and they said that they're starting to see it. And they're trying to get the divers to help eradicate it, but I think it is one of those things that once you see it, it's almost impossible to get rid of it. Like you're saying, if you break it at all,
looks like it
just releases spores. Creates more of a problem rather than less.
Yeah, so there's some organizations that are helping to note observations of these species, both sargassum.
They can barely hear you. I don't know if that's Jennifer or Annie.
Yeah, there's some a website where you can log observations of sargassum annandaria in California, because we're trying to keep tabs on if divers are seeing it other places, and so it's all voluntary. But
this is a citizen scientist website.
It's not a citizen scientist website per se. It's run through. It's called PISCO, the Partnership for Interdisciplinary Studies of Coastal Oceans. That's and the website is affiliated with UC Santa Cruz, but it's a place where you can log in and report observations, both either positive or negative, on this map, and it does help us to detect or see how it's been spreading or where it's been seen. And then others can go out and do more, like either the sanctuary or the Channel Islands National Park, say if there was another sighting at a different island in that area, and go see if they can find it, determine its extent, see if eradication's possible, that kind of thing. But just giving out information at harbors to boaters and recreational folks using, maybe renting kayaks or other things, can be useful way of increasing awareness of the importance to clean our recreational equipment and to reduce the hitchhiking potential.
When we go through the marine snow like this, it always reminds me of like Star Wars or Spaceballs when they go into hyperspeed. In slow motion.
That moment before it speeds up.
Yeah.
So why are we seeing so much blue water right now?
Because we're going down slope pretty much, and where you're trying to keep Argus off a wall.
Yeah, so whenever we go down slope like that, we wind up having like Argus has to be higher than Herc, which doesn't work out. Or sorry, the Argus is too close to the bottom, so you have to have him like higher up out of the way. Usually, we like to have Herc out front, further up slope than Argus.
Thank you.
But sometimes it doesn't always work out like that. That's why we back down hills and stuff like that.
Got it.
Oh, I thought that was so we could see the rock.
Yeah, both. So you can see, and so we don't smash Argus.
Back to the nightmare of Argus on the seafloor.
Right.
We're trying to avoid.
Yeah.
How long have you guys been using Argus and Hercules together?
Like how long have they been like on the ship? Like
Yeah, I'm assuming they weren't commissioned in 67, like the boat was, right?
Very advanced technology for the 60s.
yeah. I think from the early 2000s or
Yeah. 2002 or 4 or something like
yeah,
that.
something like that.
We were watching last week at home, and my son asked me how many dives Hercules had completed, which I didn't know the answer to, so I suggested that he write in the question, and it was answered. So at that time, I think it was 700 and, I'm trying to remember, 700 and...
Was it 1,700 and something?
I think we all start them with a 1 in front.
Oh,
I think so.
Yeah, I thought we
It's the
passed
dive
1,000
number
though.
minus 1,000.
okay.
Yeah.
Is the dive number minus
700.
1,000?
Yeah, so
Okay.
795 right now.
Okay.
Yeah.
Y2K bug or something.
So I think he wrote in on 793 then.
Yeah, that makes sense.
He was so excited that you answered him. He just was jumping up and
Y2K
down.
bug?
They answered it! And he wrote it down, and he told his science teacher at school, there's a octopus.
Yeah, how old were you?
I-
You were 6. Sorry, Y2K distraction.
Oh.
If Jess knew what it meant or not.
Just try, well, not if she'd heard of it, but to remember everybody was like, oh, the computers are all going to break. People
Yeah.
had jobs to go around and like change the computers.
Oh, yeah, totally.
I remember that.
Yeah.
And then nothing happened.
Nothing happened. Somebody got some late charges at Blockbuster Video or something.
Blockbuster what? You're aging yourself
I used to
there.
work there.
You worked at a Blockbuster?
I worked at a Blockbuster, yeah. It was sweet.
Yeah, well, those were like, if you liked movies at all and you're like younger and had to have a either
Another
part-time
octopus.
job or whatever.
It's a job with a name
Great
tag
perks.
and a shirt.
Yeah.
All right, we got an octopus to the right, left, and then to the upper right.
TVL actually
Well,
doesn't look
that's
too
too
bad.
low.
Yeah, we're
So if we can
back on the bottom.
start tracking up whenever you feel. All right, so
Somebody wants to know if you use machine learning.
We don't particularly, but people in the lab that I work in, they do. They take video transects, particularly the shallow water, and then they run those into machine learning. So they train algorithms to be able to detect different features, and then they eventually get to the point where they can just run video through this algorithm, and that will basically annotate it and tell them what types of species are in each image, and it's really quite neat. But I don't think you guys don't do much
Do they
of that.
call it machine learning or artificial intelligence?
He calls it machine learning,
And then it becomes artificial intelligence.
Okay, got it.
and also the word deep learning often gets thrown around.
Okay. I know that there's some of that being done at
I think you
UC
could
Santa
start it,
Barbara.
Aaron, and just
as part of the Monterey Bay Aquarium.
adjust it.
Marine Biodiversity Observation Network demonstration project.
MBON.
MBON.
Yeah,
That's right.
that's true. That is true.
So an example of machine learning that some of the viewers might encounter every day is when you use certain websites and it makes you identify, say, every picture that has a
No,
tree.
that's what I mean. Doesn't
or
he
every picture
settle
that has
out?
a car. That when you do
To
that,
the
you're helping
northwest?
train an algorithm. That's a machine learning algorithm.
Okay.
So we're
Well, I
very
mean-
close to the start of our next transect, and
Yeah.
so we're just trying to figure out
Sure.
that right now. If anyone's wondering what we're doing. Just
No,
hold
no,
us
I'm just start a line and deviate them over there and then cut back in. But that's okay. Just do the step and then we'll start.
When you guys say you're doing steps versus transect lines, what are you referring to?
They're busy. They're busy figuring
They're
it out.
doing it now.
In this area, we're seeing a couple different types of anemones and some white sea cucumbers.
And one of those
what is that?
Apora,
There was a
Porales,
step versus
rufescens?
line question.
Porellia
Rufescens.
I don't know if you
I
want it.
don't know. Look at the difference in color.
jelly
You gonna
though.
zoom in there,
They
video?
moved on.
It's
Moved
not
on.
that deep red. It's
Yeah,
a much
gone.
different color. Shape-wise, very similar.
Remember when they adjusted the light on the last one, though? You could kind of
Yeah.
see that inner rim. You can almost see that faintly here.
Mm-hmm.
Yeah, so on this jelly,
So cool.
you can just barely see a little bit of the oral arms way underneath for a moment there.
Did you guys want to ask Michael the question about the step?
Oh, yeah. When you guys talk about you're moving stepwise versus along a transect, what are you
What was that?
referring to?
That's hilarious. I'll give it to Aaron. Aaron's best answer.
Well,
Nope.
thank you.
This-
It's just different ways that we move the ship. So when we do a step motion, the ship just puts in, physically types into their DP a 20-meter step in whatever bearing I give them. And as the ship moves into that position, if we don't keep adding steps, it will stop. So it comes to the end of that position and stops. So it's the safer way to go if we want to make kind of short steps or we're doing a lot of sampling. When we track a line, the ship is just going to chug along until we tell them to do otherwise.
We get
Okay.
a picture with lasers for this?
Sure.
Thank you for explaining that.
Yeah, no worries.
There you go.
There we go.
You want to
Thank
face
you.
the line here,
Thank
Jess,
you.
and get all lined up?
That's pretty.
That's cool.
Oh, yeah, sure. I just want to see
They
this thing
almost
take
like
off.
to show off.
This thing.
Well,
Maybe it doesn't take off.
Wonder if they're more
Come on.
active around a light
Get-
source.
Yeah, and they can like sense our thrusters and noises.
Bearing of that line we're tracking up is 33.4.
33.
maybe it is
Roger.
gonna
So you
take
okay?
off.
We'll
Jess
start
asked
going
it
up, and if we
take
need to
off.
shift, I'll just offset it on the line.
Sure.
Sure.
You gotta
Is that
think
okay?
jellies are probably so influenced
Take
by
off,
the pressure
take off.
differentials that we're creating just from the acoustics of the submarine.
Bridge nav.
All right. Had its moment. All right, go ahead and come wide video.
Can we
That's
start tracking
beautiful.
line bearing 333?
See, there's that relaxing.
Just to repeat,
You
the
just
bearing
watch
is
that all
3-3.
day. If you could watch that all day, you would not have stress dreams at night.
No, you would not.
Yeah, you'd have dreams all day.
Oh, look, there's another one. How interesting.
I wonder if maybe
I
they're
think.
feeding in the sand.
There's a couple of them seemingly up ahead.
And bridge, let's
Three,
reduce
three
speed to 0.3 knots,
And
please.
then there's that corallimorphus anemone. I
what do we
think.
say, Aaron? 30 degrees?
No, wait.
33.
33.
What's that?
Yeah, the one with the white
Yeah,
tips.
right. Oh, was that a little eel pout down there? Missed it.
Me too.
So remember, even though you see a lot of sediment, not a lot of animals on the surface, there are a lot of animals that are living inside the sediment. So it's still teeming with life. They're just harder to see. They're smaller often, but super important.
Yeah, they estimate that the microbial life has a lot to do with the carbon and nutrient cycling. Much bigger than we anticipated in our initial estimations. And there's life way, way down deep in the sediment that can use hydrogen as a catalyst for their energy sources.
All right, we're getting close to the
Those
start
tiny
of our
little
next
stars
transect.
on the
Yeah.
left.
And what's that?
white thing?
Bright white on the right side.
Looks like a sand dollar.
Shiny. Is that a yawn I heard?
No.
No.
Nope. That was a thinking sound.
All right, I'm lined up on it.
Nice little Brazingid star. All right, are we about ready to start?
Is altitude all right for you guys, or should I come up a little higher?
I think we could come up a little bit higher. Let's be able to see a little more.
Sorry, a little bit higher.
Yes, please.
Roger.
All right, so we're going to get this transect started before our watch is done. Right?
Well, we're sure trying.
We're doing our best to get it going.
At least we got him in a good position to get right to work.
I would like to see a little forward progress. That would make me
Yeah, we
happy.
can do it. We got 10 minutes.
She's
Thanks
a
for the
positive
confidence.
You're welcome. I'm a glass half full kind of gal. We have some fun little crustaceans swimming around in front of us. That little shrimp.
Just gonna check the gauges quick, Jess.
Yeah, sure.
We want to zoom in on that coral while we're sitting here. Get a nice pretty picture.
Yeah, sure, let's do it. Go ahead, video.
Where? This looks like a little black coral. Yeah. Mm-hmm. And well, the stalked ones, but we've seen feather stars up on other animals up in the water column, in the sponges and up on some of the corals higher up. We've seen the
Is that
feather
good
stars.
on the zoom?
Yeah, that's great. Thank you.
All right, go ahead and come my video.
Even on the base of that rock, once you zoom in, you see a lot of other things, right? You see the
Yeah.
crusting sponges and other things. So if we could zoom in on every little patch of soft bottom, we'd see a lot more animals than we can see from this distance.
So when
The setup's not bad.
these videos are
You're like-
analyzed,
you're actually here, so
I-
I think it's actually
are they
not
looking?
bad.
Hello.
No. Are they mainly looking at the things that are easy, like big things that are
Yes.
easy to see, since they won't?
Video, you want to go
Yeah.
ahead and zoom on this,
That's
please?
right. Hello.
Another one of those
Good morning.
Is that?
coral.
Nice shot.
Yeah.
Yeah.
Did I hear another thinking sound up there?
Yes.
Oh, absolutely. So you're going to start to
You're going
hear
a
some
little tighter.
conversations in here as
Yeah.
there's a shift change.
Too much shadow.
On my left, I have a transition between data loggers, where Magda is sharing her information with Taylor Ann on the things that we've done this dive. And on the right side, I have Jennifer sharing with the lead scientist, Erica, about the transects that we've completed and what the plan is for their shift.
Got an enemy.
Is he a friend of the coral or an enemy? Mon ami?
All right. Go ahead and cut my video.
So they're a little octopus.
Yeah. Kind of with the more curled legs like a female. Ish.
Yeah, it's hard to tell.
Yeah.
You can't.
Go ahead and zoom in on their video.
So we've started this line.
Yeah, so we're
Just-
on the transects. Just ship's not moving yet. I don't know if you want to mark it as start of transects.
Our data is switching out. So.
Okay.
Go ahead and cut
Keep
my
on
video.
second.
Come on.
Somebody just wrote in, with fronds like these, who needs anemones? Well played. Keep your fronds close and keep your anemones closer.
Ooh.
Ooh,
Hey,
Go
look
ahead
at
and zoom in
that.
on there.
that's cool.
Is that a jelly?
Looks like it.
Looks like a jelly.
Cool.
Ah.
Morning. Try to tell you what that jellyfish is that we're looking at. See if it's in our identification guide.
A gel anemone.
Gel anemone.
Yeah, kid.
I
Go ahead and cut my video.
it possibly is related to an Agenura, but this one looks like it has
Full wide video.
a little more
Thanks.
of those spicule-like things and a little more uniform color in the top. So I speculate it's something that isn't in this identification guide. My replacement is here, so I will say goodnight. And you can say hello to Amanda.
Say goodnight, Jennifer. All right. Well, as Jennifer and Annie mentioned, our replacements are here. So thank you for joining us. This is Science Communication Fellow Scott Simon. I'm going to sign out and turn it on over to Danny, who will be coming up in just a moment. Continue our transects in the Monterey Bay National Marine Sanctuary on the Davidson Seamount. Good night.
All right, good morning.
Good morning, everyone.
Good morning.
Got a watch change. So our science lead is Erica Burton from the Monterey Bay National Marine Sanctuary. And I am Science 2. My name's Amanda Kahn from Moss Landing Marine Laboratories.
And good morning, everybody. I'm Taylor Ann. I'm the Ocean Sciences intern. Let's see what we have in store today on this transect.
Yeah, just a quick science summary. We've been running transects across a small feature, the top or center of which is where we've been seeing these really dense aggregations of octopuses brooding eggs. The octopus species is Mus octopus robustus, and they are there in clusters of up to approximately 30 to 40 octopuses in a single space. So it's really kind of neat, really exciting to see them all, and we want to figure out the extent of those brooding aggregations. And so we've been running transects all night across this feature to see the extent, horizontal extent of these octopuses. So we are running along our final transect line right now of this dive. And as our watch shift is completed, then everyone will tune back in and we'll be underway heading down on this transect line. Right now we're hovering as everyone's figuring out, getting their bearings, and learning whatever they need to know from the previous watch to plan for what's coming up next.
Hello, explorers.
Sorry.
Welcome to Nautilus
Are we on
Live. This
transect?
is Danny. I am your science communication fellow. I see so many of you guys are watching us. Thank you for joining us for our last transect of this dive, but don't you worry, we will have more dives to come. We've got at least one more. I think it's another 30+ hour dive. So you guys will get a chance to see plenty more things as we explore Davidson Seamount here.
Let's see, what are we seeing this morning?
Ooh, a little bit of sponge in the morning. So it gets me up. Gets me up in the morning. Sorry. Yeah, we see several different types of sponges in this view. So all that I am seeing right now are glass sponges of the class Hexactinellida. So these guys are generally sponges are filter feeders, and class Hexactinellida is one group of sponges. It's primarily found in the deep sea. These guys make their skeletons out of glass. They extract silicon and oxygen from the water and make silicic acid and deposit it around some proteins to make these glass rods in their skeletons. So it's pretty cool. These guys can filter huge amounts of water. It's about 900 times their own body volume every day is pumped through their bodies. And from that, they eat bacteria. And that's pretty neat because other animals aren't able to tap into bacteria as a food source very easily. So sponges are tapping into this food resource. And by turning it into sponge or sponge poop, they're able to make food that is edible for the animal food web. Also in view here is a black coral. So that is a coral of the genus Bathypathes. So that's one coral, but it has many individual polyps growing on it.
It looks like a feather.
It does. It looks like a beautiful feather. Can you tell us a little bit more about sort of how a coral works? Like its skeleton, and then you said it's a colony?
Mm-hmm. Yeah, so a coral builds a skeleton that then the live polyps are growing on. So for these corals, these are octocorals. So they have actually an internal skeleton, which means they have tissue on the outside of it. And then on that skeleton, if we could do a quick zoom, you guys can see all of the individual polyps.
When she asks for zoom, you can go ahead.
You okay?
Yes.
Okay.
Yeah, I'm okay.
Zooming in.
Great. Thank you. So you can see the individual polyps on those arms of the coral.
And our polyps are the ones that are sort of making the food? Or that's the living animal?
That's the live tissue.
That's
yeah,
the live
and those
tissue
are
of our coral.
the feeding parts of the coral.
Beautiful.
Yeah. Thank you. Oh, that's a great view. Okay, thanks for the zoom.
Okay, coming wide.
So the way corals eat is those polyps all have stinging or ensnaring cells
Full wide.
called cnidocytes, and those have nematocysts inside. And the nematocysts are kind of like barbs or harpoons.
Copy that.
Oftentimes they have a type of venom in them that can sting. And so as prey, like small crustaceans, drift by, those crustaceans will get ensnared by those harpoon-like nematocysts and trapped against the coral, and then the polyps can draw them to the mouth and eat them. I don't know what this particular species eats. If there's
I don't either.
Yeah, if they eat marine snow down here, or
and we see more of these black corals at the deeper depths of the seamount. On the top of the seamount, we see things like bubblegum coral that can be 3 meters, 4 meters tall. But down in the deeper sections, we see a lot more black corals. I'm not sure why. Maybe they do collect snow. That's a cute sponge.
Thanks for the view
Getting
of
one's
this
home
guy.
inside the tulip.
Sure. Thank you.
Is that a brittle star on its outside?
Oh yeah, we got a brittle star on the outside. This it's a stalked glass sponge, and down at the base I can see those little tendrils down there are part of a crinoid, which is a type of echinoderm like sea stars and sea cucumbers.
So those are hitching a ride to get up into the water column, you think?
Yes. Yeah, so by climbing up on that stalk of the sponge, they're higher up and able to have more food drifting by. So crinoids are suspension feeders as well. The more water that goes by, the more food they get to eat. Thank you for the zoom.
Okay, coming wide.
Did you call that a tulip sponge?
I called it a tulip sponge.
That's a good name.
We don't really have common names for deep sea sponges, so.
It is pretty. It does look like a tulip. A little rosebud, something.
Rosebud.
Rosebud sponge.
So we're moving along a transect line. Throughout the night we've been, there's my sponge.
Where?
Up on the top of that rock.
Are we waiting for the ship to catch up?
Yes.
There's a neon color sponge at the top left.
Greeny yellow.
That if we have time, it'd be great to pick that up.
We could slurp it. They're very soft and floppy.
I would say we got time for a slurp if you want.
Fantastic.
The yellow thing there, right to the...
Sort of center screen almost?
Yeah.
Yes, if we could get some nice close-ups of it first, that would be great.
You want to slurp it, do you?
Yes.
Go get it.
Nice size, too.
It is.
It's so cute.
Sorry, I was pushing buttons.
So
okay, go ahead.
Yay. So this sponge was a mystery sponge for me last time, and we collected a small piece, but I haven't figured out who it is yet, so having another piece is going to be really helpful.
It is such an interesting color. Do we have any idea why it's that yellowy color?
Not a clue.
All right.
Yeah.
Mystery of science.
Yeah, exactly.
Well, Amanda, you are a named a favorite scientist. So
That happens?
Willie, yes,
Guys.
absolutely. We have fantastic viewers here at Nautilus Live, and it says, Amanda, you are my favorite scientist
give
in
you
the
the,
crew.
other
You
camera
introduced
too.
me to my new favorite organism, the xenophile-phores.
Oh, no way.
Can you,
The
can you
previous
go wide
crew
wing
talked a
so
little
help
bit
her
about it,
get
how
the
it eats. Its diet is marine snow.
suction sampler?
But I want to know more about this amazing, massive unicellular organism.